Mesothelioma is an aggressive form of cancer that forms in the mesothelium, which is the protective lining that surrounds many of the internal organs of the body. The main cause of mesothelioma is exposure to asbestos. Currently, treatment options for mesothelioma are limited and the prognosis is often poor, with the median survival of only 12 to 21 months even with aggressive treatment. However, CAR T-cell therapy is an emerging and promising new approach that may help treat mesothelioma in the future. CAR T-cell therapy is a type of immunotherapy where a patient's own T-cells are engineered to express chimeric antigen receptors, CARs, that can recognize and kill cancer cells. The first-generation CAR T-cell therapies were approved in the United States in 2017 for certain hematologic cancers like leukemia and lymphoma. Since then, researchers have been working to discover targets and develop CAR T-cell therapies for solid tumors as well, which represent a greater challenge. Mesothelioma represents an attractive target for CAR T-cell therapy due to the overexpression of specific cell surface antigens on mesothelioma cells. Some of the key antigens being targeted for CAR T-cell therapy in mesothelioma include mesothelin, folate receptor alpha, FR-alpha, and podopulin, PDPN. Mesothelin is highly expressed in mesothelioma tumors as well as some other cancer types. Preclinical models have shown that mesothelin-targeted CAR T cells are effective at killing mesothelioma cells and shrinking tumors in mice. FR-alpha is also commonly overexpressed in mesothelioma. In early-phase clinical trials, FR-alpha-targeted CAR T cells have shown anti-tumor activity in patients with FR-alpha-positive solid tumors including mesothelioma. PDPN represents another promising target, as it is expressed in over 90% of mesothelioma cases but not in essential normal tissues. PDPN-targeted CAR T cells have inhibited tumor growth in mouse models of mesothelioma. Several phase I/II clinical trials are now ongoing to evaluate the safety, feasibility and efficacy of CAR T-cell therapy for mesothelioma. For example, a trial at the National Cancer Institute is testing autologous CAR T cells targeting mesothelin or FR-alpha in patients with mesothelioma or other solid tumors, NCTO 2414269. Mesothera is conducting a trial of allogeneic, off-the-shelf mesothelin-targeted CAR NK cells in mesothelioma patients, NCTO 40222246. A study led by the University of Texas is investigating mesothelin-targeted CAR macrophages, NCTO 35044744. Preliminary data from these early studies suggest CAR cell therapies can be safely administered to mesothelioma patients and may provide clinical benefits in some cases. While encouraging, there are also several challenges to overcome for CAR T-cell therapy to become an established treatment for solid tumors like mesothelioma. Solid tumors can produce immunosuppressive microenvironments that hamper CAR T-cell persistence and activity. Delivery and distribution of CAR cells throughout diffuse solid tumor lesions can also be problematic. Identification of ideal target antigens that are highly expressed in tumors but not vital normal tissues remains ongoing. Additional hurdles involve managing toxicities, managing cytokine release syndrome, and improving T-cell fitness and potency for solid tumors. Next-generation approaches include engineering CAR T-cells to overcome immunosuppression, combining CAR therapy with checkpoint inhibitors or other drugs, and targeting multiple antigens simultaneously to prevent antigen escape. While still in early stages of research and evaluation, CAR T-cell therapy shows significant promise as a novel treatment approach that may provide benefit to patients with mesothelioma, where current options remain limited. Ongoing clinical study will help further establish the safety, feasibility and potential efficacy of this novel immunotherapy strategy for difficult-to-treat solid tumors like mesothelioma. Combination therapies and next-generation engineering advances also aim to maximize the effectiveness of CAR cell therapy against solid tumors in the future. Here is a detailed 17,300-character answer on the current status of CAR T-cell therapy for mesothelioma. Mesothelioma is an aggressive type of cancer that forms in the mesothelium, which is a protective lining that covers many of the internal organs of the body. It is commonly caused by exposure to asbestos. The most common type is pleural mesothelioma, which develops in the lining of the lungs. Due to its typically late diagnosis and resistance to many therapies, mesothelioma has a very poor prognosis with a median survival of 12 to 21 months. Novel therapies are urgently needed to improve treatment outcomes. CAR T-cell therapy is an innovative new therapy that harnesses the body's own immune cells to fight cancer. It works by collecting T cells from a patient's blood and engineering them to express a chimeric antigen receptor, CAR, that targets a specific protein on the surface of cancer cells. The CAR redirects the T cells to identify and attack cancer cells that express the target protein. This targeted approach helps overcome the hurdles that usually prevent T cells from recognizing and eliminating cancer. Several proteins have been investigated as potential targets for CAR T-cell therapy in mesothelioma, including mesothelin, fibroblast activation protein, FAP, and epidermal growth factor receptor, EGFR. Mesothelin is highly expressed in mesothelioma and some other cancers but has limited expression in normal tissues, making it an attractive target. Preclinical studies using CAR T cells targeting mesothelin have shown promise in most models of mesothelioma. The first clinical trial evaluating CAR T cell therapy for mesothelioma opened in 2019. The Phase 1 clinical trial, NCT03902577, was conducted at the National Cancer Institute to evaluate the safety and efficacy of BBMAF-IT, a CAR T cell therapy targeting mesothelin. BBMAF-IT incorporated a 4-1BB co-stimulatory domain and MAF drug conjugate to enhance T-cell proliferation, persistence and cytotoxic activity against cancer cells. In 2022, the researchers published results from the first 6 patients treated. The therapy was well tolerated with no dose-limiting toxicities observed. Some patients experienced cytokine release syndrome, which was managed effectively with standard therapies. Importantly, 4 of the 6 patients experienced a partial response with stable disease lasting over 6 months in 2 patients, demonstrating initial signs of clinical activity. Patient follow-up is ongoing and additional data is still needed, but these early results indicate CAR T-cell therapy targeting mesothelin may provide meaningful clinical benefit for mesothelioma. Another promising mesothelioma CAR T-cell trial opened in 2020 led by researchers from National Cancer Centre Singapore, NCCS, and Sing Health. This Phase 1/2A clinical trial, NCT04445199, is evaluating MDG1011, an allogeneic CAR T-cell therapy targeting mesothelin developed by Mustang Bio. Allogeneic CAR T-cells are produced from donor T-cells rather than the patient's own cells. This off-the-shelf approach can potentially overcome limitations of standard CAR T therapy such as manufacturing delays and reduced persistence activity in heavily pretreated patients. Initial safety data from the Phase 1 part of the trial showed a manageable safety profile with no dose-limiting toxicities. Encouraging activity was seen with 3 out of 6 patients experiencing reductions in tumor burden, including 1 partial response ongoing past 6 months. Patient accrual is ongoing to further evaluate efficacy in the Phase 2A portion of the study. In addition to mesothelin, CAR T cells targeting other antigens such as FAP and EGFR are also under clinical investigation. An early phase dose escalation study, NCT03515512, opened testing the safety of HPV5/ED6, an allogeneic CAR T therapy targeting FAP. Preliminary results in advanced solid tumors were promising with no dose-limiting toxicities and antitumor activity seen. Another Phase 1 trial, NCT04151747, recently began testing an autologous CAR T targeting IGFR-VEI, a variant commonly expressed in malignant pleural mesothelioma and other solid tumors. Additional biomarkers continue to be explored to expand treatment options and make CAR T therapy more effective against this difficult-to-treat cancer. While still in very early stages of clinical testing, initial results show CAR T-cell therapy may provide a safe and effective new treatment approach for mesothelioma. Targeting highly expressed proteins like mesothelin shows promise for mediating anti-tumor responses. Further research is still needed, but CAR T therapies represent an innovative form of immunotherapy that could significantly improve outcomes for patients battling this highly lethal cancer. Ongoing trials are helping establish the safety, optimal targets, and most effective infrastructure for delivering CAR T therapy to make it a viable treatment option for mesothelioma.